Meiosis is the cell division that occurs in sex cells, and the process reduces the starting diploid cell, resulting in 4 daughter haploid cells, and reducing the chromosome number in half. In meiosis 1, the homologous pairs separate, and in meiosis 2, the sister chromatids separate. A replicated chromosome has 2 identical DNA double helices and is made from an unreplicated chromosome in the S phase of interphase. All our body cells contain the same mix of DNA. 46 chromosomes grouped into 23 pairs, 1 in each pair from your mom and 1 from your dad. Those pairs of chromosomes are similar but not identical because they contain different genes or alleles in the same spot for any given trait. Because they are similar, they are called homologous chromosome pairs. There are some special cells that have only 1/2 of that amount, 23 chromosomes. Those are sperm and egg cells. Those are haploid cells. They have half of full sets of chromosomes, and they need each other to combine to make a complete 46. To create those kind of cells, a process that is very similar to mitosis is required. This process is called meiosis. In mitosis, 1 diploid body cell ends up into 2 diploid body cells, whereas in meiosis, 1 diploid body cell divides in half, twice, into 4 haploid sex cells. Meiosis goes through the same stages as mitosis: interphase, prophase, metaphase, anaphase, and telophase. But then it goes through another cycle of those stages again: prophase 2, metaphase 2, anaphase 2, and telophase 2. To start meiosis 1, we begin with interphase 1. Strings of DNA in the nucleus begin to duplicate, leaving 2 copies of every strand. Now we begin the PMAT cycle, similar to mitosis. In prophase 1, centrosomes start heading to their corners of the cell, unspooling the microtubules, and the DNA clumps up with proteins into chromosomes. Each single chromosome is linked to its duplicate copy to make an X-shaped double chromosome. Each chromosome is called a sister chromatid, 1 on each side of the X. In prophase 1, crossover and homologous recombination happens. In meiosis, every copy is going to be different from the rest due to crossover. For crossover to happen, each double chromosome lines up next to its homologue and form a tetrad, and they trade sections of DNA wherever they come into contact. That's called recombination. The sections that they're trading are from the same location on each chromosome. One is giving up a genetic code for something else. Since all 4 chromatids have traded some DNA segments at random, all 4 chromatids are now different. Now there are 22 pairs of chromosomes, and there's 1 pair that doesn't go through crossover or recombination. They are sex chromosomes. For females, they have 2 X chromosomes where they can cross over themselves because they are the same. Male get 1 of those X chromosomes and another from your dad, the Y chromosome. During prophase, the Y chromosome doesn't do anything because they are not homologous, because the XY pairs on these chromosomes will split later on into the chromatids. Half of the 4 resulting sperm will be X and half will be Y. Next is metaphase 1, where all the chromosomes line up in a single row next to its homologous pairs, and in anaphase 1, the sister chromatids get pulled apart and moved to either end of the cell. Variation occurs in this due to independent assortment. It is the process of random segregation and assortment of chromosomes resulting in the production of genetically unique gametes. Finally, telophase 1, where the nuclear membrane reforms and nucleoli form with them and the chromosomes change back into chromatin. Then cleavage forms between them, and now as the 2 nuclei move apart from each other, the cells separate into 2. Now we have 2 haploid cells, each with 23 double chromosomes. In these new cells, the chromosomes are still duplicated and connected at the centromeres. They still look like X's. Now as it goes through the phases 1 more time, the goal is not to duplicate the double chromosomes, but instead to pull them apart into separate single-stranded chromosomes. Because of this, there is no DNA replication involved in prophase 2. Instead, the DNA just clumps up again into chromosomes, and the infrastructure for moving them, the microtubules, are put back into place. In metaphase 2, the chromosomes are moved into alignment into the middle of the cell, and in anaphase 2, the chromatids are pulled apart into separated single chromosomes. The chromosomes uncoil into chromatin, and the crease forming cleavage in the final separation of cytokinesis, which is the end of telophase 2, results in 4 sex haploid cells.